Navigating the combination is about the real decisions in combination product development, the trade-offs, the judgment calls, and the things that don't fit neatly into a guidance document.
Each episode, we sit down with leaders from across drug delivery to talk through what's actually changing and the strategic challenges of bringing a drug device combination product to market.
This is what we've learned. Let's help you do it right the first time.
Hello, everyone. Welcome to today's episode of Navigating the Combination. I'm Jonathan Amaya-Hodges, joined by my co-host Parth Kathari. Today, we have Matt Wills from SE02.
We had a great conversation with Matt, where he shared what it looks like to operate at the intersection of primary packaging innovation and combination product development, and why a system-level view of drug-device interface is becoming essential for both pharma and biotech teams.
We hope you enjoy. Hi, Matt. Welcome to the podcast.
Jonathan, thank you for having me on here.
Great. I'll open things up here. I understand you spent your career at the intersections of material science, primary packaging, working with the containers that drug products actually live in. Before we get into specifics, help us frame the conversation. What is it about newer biologics, gene therapies, that's pushing the limits of traditional glass and plastic? And what does that matter for combination products?
Well, I think on the formulation side, the industry is moving away from some robust molecules into increasingly sensitive molecules. We're talking more targeted therapies, complex suspensions. And so as the sensitivity increases, the container is no longer playing a passive role. It's not essentially just a vessel. It's becoming much more part of the system. And that's both from a regulatory push over the last decade and also as from a risk management profile and the ownership of those risks.
Excellent. And where do you see glass kind of hitting its maximum as the kind of traditional container and plastic or hybrid approaches picking up?
I think it's a lot of it has to do with that sensitivity overall and what is the nature of the interaction. I mean, we always talk a lot about the bulk material, but what really is the focus is what is the drug contact surface itself. And so the bulk material might be glass, it might be plastic, but what the drug contact surface is really what's interacting with the drug, whether or not you're getting interaction with the particles, subvisible oil, whether or not there's adsorption, and how that interaction creates any leachables, extractables coming from the bulk material. I think that's where we're seeing the sensitivity coming out from a glass side in some unique aspects that branch into higher pH as well, going into risk of delamination from a glass side. And so there's kind of just becoming less of a one tool fits all option from a glass side. And so there's been much more approach and expansion into plastic and other hybrid materials.
Mm-hmm. And of course, there are pros and cons to almost everything in the world. So what challenges might remain even with plastic or hybrid types of approaches?
So pros and cons from glass. I mean, you're talking about it's robust. It's been in the industry for 50 plus years or so, 100 years. It's very well characterized, very well known. Cons of it being is it's limited mainly from a device design, right? You have limitations in glass formation products to a drug assembly itself. The boundaries, again, pH, going to a very high pH, you're talking about 10, 11, 12, you're risking delamination issues. And so then that's just a great combination compatibility issue right there. And then at the same time, leachable and extractable profiles, they're known, but are they suitable for the drug environment that you're interacting with, being from tungsten, from glues, from the needle adhesion, if you're looking from a state needle assembly, or the metal ions within the borosilicate glass itself, because we always think of glass as just glass, but really it's a borosilicate glass. And so it's more of it's melted sand at the end of the day. Very clean, but it's still melted sand. So I think that's where we're running from the pushes of limits there. But on the straight switch to a glass side, plastic side, you're losing some trade-offs from bulk materials. You might still maintain, depending on your plastic material, just wide varied options, but most of them you lose just an oxygen barrier performance between them. So if there's a gas barrier issue with oxygen, nitrogen, or sterilization compatibility from ET or NO2, or is a new sterilization method that's coming out. But you do gain the advantages of a lot more complexity, a lot more design controls of just the device design itself. So you can be more accommodating to device integration, to auto injectors, on-body pumps. So that's kind of where they see the advantages of the physical plastic side, breakage in there, but it's kind of not having all of the benefits from the glass keeps the transition from going into a plastic product. But the limitations around the glass side and the advanced formulations just can lead to a forever ending of constantly trying to redevelop a formulation to fit a static system.
That's quite interesting. So, when you're evaluating like any container system, right, whether it's glass or plastic or hybrid, you mentioned that the drug contact surface is that first critical parameter. Could you maybe walk us through like how you think about the drug contact surface and what like drug product teams need to consider when they're evaluating that compatibility with the container?
Yeah, so first things first, inorganic versus organic. Glass is being a more organic side, plastic is inorganic. So that's one big difference right there from material interaction, surface free energy that leads into hydrophobicity, glass being very hydrophilic, plastic being very hydrophobic. In some cases, I've seen that's where actually it's a great benefit for being hydrophobic from like residual volumes left over after dosing. Some cases that it can lead to also then an adsorption to the surface that's unwanted because then there's a loss of efficacy of the drug formulation. And then to bring in hybrid materials, you're talking about really talking about trying to bridge a balance or really design and tailor a surface that's more suitable in both realms to where it's much more of a bystander to the system, very inert, very non-reactive, but still has certain properties that basically maintain formulation balance.
So I can imagine, right, when pharma teams are developing these drug device combinations and they have this interaction between the container and the drug product, what might be some biggest misconceptions that maybe they have about what's like actually happening at that surface between whether it's during the period of storage, during the period of administration? What's something that you can see or you constantly maybe
So I think one
encounter?
big thing is treating the syringe or the device as a single component. I think it's keeping out, forgetting the fact that it's not one large component. It's a multiple component itself. You got the syringe barrel, of course. You have the lubrication itself, which is going to have a different role in that surface interaction. You have the plunger, which can have its own interaction on a different side because it's typically also in drug contact with the system. And then they have if you have a stake needle, you have whether or not drug is in the needle at the time during the storage, or if it's a lure in, how it fits into the lure, and then that transition from a lure to an add-on needle. All of those can play a different role in the drug interaction. And so it's kind of what we typically try to do is take a step back and say, away from saying, okay, it failed, but more of understanding what is the mechanism going behind. And so that way you can start tracing down to components of the syringe to get a better understanding of the mechanism. And through testing and through root cause investigations, that's where we kind of push a little bit more new methods in terms of how do you set up your testing in maybe a non-traditional way, but in a way that isolates different variables of the product. You know, if you're trying to look at just the plunger sealing within the syringe, do you have to have the whole needle system beyond? Or can you cut above the plunger and isolate that in some way that you're effectively only measuring that plunger interaction in order to reduce any other variables from interacting with your test setup? I think that's kind of what we've looking with these conversations is moving towards what is the actual mechanism, what's triggering it. And it's not always just the syringe. Sometimes it's the siliconization of the syringe. So the syringe is fine, but it's the siliconization method itself.
I'm really glad you brought up siliconization because that's bringing me some memories from my time working on pen injectors, auto injectors, wearable injectors, and silicone oil migration was always like a huge hot topic, right? And so sometimes it's considered like a positive feature, sometimes it's considered a problem. I'd be curious how you help customers navigate that trade-off because I know that was something I encountered quite often.
Yeah, I think so silicone oil, it solves an engineering problem, right?
Sure.
You need to get your drug delivered effectively, consistently, reliably. But it has its own risks, and I think the large part first is trying to understand what is the risk that is introduced from silicone oil, and is that acceptable for this particular molecule? There's a wide range of, at least from a molecule interaction, that there isn't no risk. And so just a traditional siliconized, not even going to a baked on silicone or even a triboglide or other kind of lubricant systems has no issue. It's when it's less of silicone oil itself. It is what is it about the silicone oil and what is the problem you're looking at? Because also when we talk about migration, is it the interaction with the drug, or is it the changing or affecting of your break loose glide forces over time, your aging profile? So with silicone oil, the question really is kind of what is the risk that's being introduced into your system and how acceptable it is that with your particular molecule. Migration is a big question around silicone oil and whether to create silicone oil subvisible oil, silicone oil like droplets for particles. Those can and cannot be a different risk assessment based on the molecule you're looking at, how sensitive it is from protein interactions, or especially from ophthalmic interactions. I think there's two directions around, or two responses to silicone oil. One is, how can we just make sure we minimize its interactions? Because we've known it, we've used it for 50 years. It's if it was harmful, at least to the human body, like we would have certainly found out proof of that. The other side is, can we just stop using it? I think that's the big part push on the other side is can we find something else that's just to reduce one, and that's not necessarily from a risk to a human or risk to a drug. It's one less variable that has to be assessed, interactive, design controlled around, and mitigated. And so that's where we see often these silicone oil-free systems, alternative lubrication methods, Teflon coated plungers, those kind of other drug delivery systems that kind of as alternative ways to remove that risk. But at the same time, you're just shifting your risk from one area to another one, but maybe it's an easier risk to control for your formulation.
Great. Thank you. I've also learned to appreciate the importance of silicone oil during my career, starting really from the beginning all the way up till now. So really good points. Maybe shifting a little bit in terms of what you and your company do right now. There are a few different strategies for addressing the limitations of glass, including multiple layer plastics, plasma treatments, and what you guys do with your coating technologies. Help us understand what makes the approach different and what it's solving for.
So the approach different here. I mean, it come back to where I said earlier, where it's trying to separate out the difference between the bulk material properties and the surface properties. The technology specific in this case that's being used in this company, it's PECVD. So you're adding a new layer to the inside. So it's a multi-layer like system, but the layer is in a nanometer level thickness. So you're essentially minimizing its overall effect to the bulk system, and more just focusing on allowing you to kind of engineer that drug contact surface independently of the whole structural container. And there we think get down into with the technology, you can start looking at how does that impact from a lubricity profile? How does that look at your barrier performance? What are the things that you lost in terms of switching from a glass container to a plastic container that were critical to the whole system that you can now try to bring back in engineering at the point of use rather than as just the whole material itself. And that as a part of that, what it creates is a lot of design flexibility where you're able to, in a sense, the drug can stay nestled in a glass-like container, but to a user, to a fill finish company, to the design of auto injectors and assembly devices, you have that flexibility that you get back with the COP device. And so that's just one avenue that we've taken from approach to kind of engineering that interaction to add new options to the market rather than just having, do you want glass or plastic as a simple answer?
Interesting. And of course, like we were talking about earlier, there's trade-offs. So where does your technology really shine? Where does it have some caveats that need to be planned around?
So one of the big one right now is regulatory risk. Of course, it's a new product, it's a new technology, it's a new interaction. So there's a lot of assessments that needed to be done, and that fortunately have been done over the years from different toxicological studies, leachables, extractables, direct drug product API stability studies for 2-3 year shelf life. So there's been a growing data behind it, supportive, but it's when you compare that to 50, 100 years of glass regulatory data, it's a drop in the bucket, it can feel like, from working with pharmaceutical companies to take the risk from a new container. And there where we've slotted in best is not being a glass replacement, but being from a fresh launch as an alternative option to evaluate on the early stages of development, not as a whole end of life cycle swap.
Mm-hmm. Makes sense.
Something I've appreciated from our conversations that we've had before is that you're thinking about it as a system, right? Not necessarily as just the barrel, but the whole device. And one of the things that I've been thinking about a little bit is that sometimes you can have that perfect barrel coating for glass barrier performance. But if, let's say, the interaction that you have with the lure tip or the rigid needle shield isn't secure, it's not strong, and you have some sort of challenge there, the whole benefit of what you've selected kind of goes out the window. So I'd be curious how, when you're working with different partners, how you bring that system level thinking to them when they're evaluating all of the components, maybe in isolation, and then it comes all together at the end.
Yeah, I mean that's very true. The system changes chain as weak as the weakest link. Same with the system. It's going to perform at the level of your weakest component, especially if you're talking about gas barrier system. If you need a very gas tight system, you don't put an airtight rigid needle shield. That's a hole in the bucket from trying to keep the air out, essentially. And that same goes from, and what we've seen, at least from a lot of different of these components, they're all not one company makes every component to a syringe. You know, you have the syringe manufacturers, you have your needle manufacturers, your plunger, your rubber material manufacturers. There's a lot of optimization that's done that's actually, it's largely independent. There's collaboration studies that are put out, but the initial part is a lot of them, they have a common goal, but they're done independently. And so that becomes our approach when talking to pharma customers from evaluations, is to get early upfront to have the conversation of what systems have they've already evaluated from a drug interaction relationship. Maybe they know a certain rubber formulation is incompatible, be some kind of a material profile from that. And to go ahead and have those upfront to go ahead and demonstrate what compatibility data has already been driven and already been created. So early on, we would do a lot of sampling from every plunger under the sun in case for break loose glide forces, just to have that full range of when they come to us and say, at least for the bare minimum of break loose glide forces, be compatible with your system. Because you can say it's ISO all day long.
Sure.
That doesn't necessarily mean it performs equivalent between each of them, right? A West plunger, Nova Pure plunger, and a BD Fluorotech plunger, their rib designs naturally create different break loose glide forces. They might age at low races, but one might be a couple newtons higher in a glide force, and going ahead and trying to understand how is that going to be impactful to your system. You know, is it from a pre-filled syringe? So then it's more of a user operation. Maybe those 1 or 2 newtons matter. Or if it's an auto injector, is that just a different spring design? And is that spring design an easy fix to hot swap between your design development? It's about having those questions more upfront to maintain a level of flexibility as your design move through your design controls. Because typically, I mean, you're trying to lock down your design, but you don't want to lock down to a point where you have to make a critical change and take 20 steps back. So it's trying to we try to adjust the approach to have that flexibility long-term, but with full understanding and knowing what that risk profile looks like.
Yeah. You can't even forget about container closure integrity, right? That's what matters at the end of the day. And so where do you see that container closure integrity risks get underestimated when pharma partners are selecting their primary packaging?
Yeah, I mean, CCI it's not nice to have. It is a pass-fail requirement. It's you have it or you don't. I think some of the bigger risks we've seen is, if we're looking at specific to plungers, is the material selection. You know, there's a lot of push from a lot of their options for moving away from silicon oil is going into Teflon-based plungers. Great friction forces because it's Teflon, but the CCI isn't quite there, especially at a cold storage, because the shrinkage rates and then how the film, whether it's a film or a fully molded Teflon, it's a different material. And it's kind of one of those, is it going to really fit your system? You know, if you don't have to do cold storage, or you don't go through what is the actual use of the drug product and it's going to see, if you can maintain that in a way that maintains your CCI, even though there's higher risk, then that's a route that you can go for, because that's the risk profile you've understood. But that's not necessarily, you know, a hot swap plunger that can make it go that way. And then that's also a big difference between glass and plastic. Is the reason why glass sometimes has higher risk for CCI on cold storage is because the material expansion rates are different between glass and plastic. So the plastics can actually shrink and expand at a similar rate, at least a much more similar rate to the rubber material than glass can. So it actually can maintain CCI through the shrinking and expanding of freeze-thaw applications.
Interesting. Thank you for the explanation there. In our prior conversation, you raised an idea that I haven't really thought of before, which is using the same surface technology across API, storage, tubing, vials, and the final container as well. We've been talking about the final container for the most part. And so if you build like for like across the components, that might aid you from a regulatory and validation standpoint. You know, what have you guys really done in that space, and what would it take to get to kind of a complete manufacturing system like that.
So yeah, one of the things there is when you're moving from your back to the drug contact surface, when you're moving from one drug contact surface to another surface, typically if you look at what the formulation goes through and from when it first manufactured to when it finally ends up in a syringe, it doesn't just see the same glass surface, the same plastic surface. It can go through PETG bottles, PET bottles, Teflon tubes, the whole mix shebang. And one of the routes that with trying to get those as similar surfaces, be it through a PECVD-based technology or a material choice, is if you can make the more common surfaces more common, it's just reducing the risk profile. And that there helps you at least helps from the better understanding of what your validation steps you're gonna have to go through from each stage, minimize the risk. Maybe you can also then build on to bridging studies, have more leverage behind that end to shorten or better rely on accelerated study rather than having to wait a full real-time stability aging study. And I think those are the goals that we end up going to. So it's not necessarily eliminating validation, it's eliminating much of the unknowns, which then helps ensure a successful validations as you move through.
What kind of validation and regulatory work would a company need to do if they were to make that argument common surface across a drug contact chain? And what might be a realistic bridging argument that they could potentially make?
So they would still need to go through the full kind of a primary container surface interaction validation stroke, just like you would go through for a standard vial. It'd be similar to the lines of a transition from vial life cycle change going into a pre-filled syringe. There, the goal is, so it's that similar level of work, but with the goal of common surface technology is to minimize the level of extra understanding going on. Because if you're going from a vial to syringe, you're adding in this case, silicone oil, right? So if you're able to say, I'm going from a 2 ml vial to a 10 ml vial, that's the kind of regulatory pathway you'd be looking from an argument of saying, well, I have a 10 ml vial, and it's being pulled from a 20-liter container, being able to treat that as going from a small vial to a slightly bigger vial because the overall surface is maintained the same way. How it's being handled and stored is maintained the same way. So it's not actually interacting with the new drug. There's still a level of due diligence that would be to prove out, but that becomes more on, I think that it's going to be, it's more pushed on the technology. And once you prove it 2 or 3 times, then you're more looking to how do I prove like for like from one container to another rather than having to reprove drug interactions performance, So then it's more focus of shifting away of will the drug perform the same way in a 20-liter versus a 100-liter API container, and more of how do I just prove that the material interaction between the 20-liter and 100-liter are equivalent because I already fully understand how my drug is going to interact with that surface.
So to close us out here, I have a little bit different question, kind of from your perspective. But you've spent over a decade watching the primary container space evolve with new materials, new coatings, new regulatory expectations, new drug modalities. What would you tell someone just entering the space today that you wished had been told to you when it started?
Yeah, I think it's kind of what we're talking about earlier from the device versus systems. I think early in my career, I thought that success just come from fully understanding and mastering an individual component, becoming that subject matter expert of a rubber or of a needle shield or of a syringe barrel. Now what I've learned is that the actual success when we're talking about going to a market commercialization is the system itself, not just the component. So the further you progressing in this field, the less important that individual component knowledge becomes and more becomes the interfaces between them. So it's not having to be the deep level expert on every single component, but because many times you'll never be the smartest guy in the room when it comes to a plunger design or a barrel design. But the interaction between the two is the gap that's often missed because most of those are also built in isolation. And then it's in the interaction that's kind of left off the assembly device builders that have to then make sure that they behave well together.
Thank you. Thank you. Excellent. Good conversation today. We really appreciate you joining us.
No, thank you for having me guys on. It's been a pleasure.
Thank you, Matt.